So our next speaker is Associate Professor Michael Stark. Michael Stark is a Senior Staff Specialist in Neonatal Medicine at the Women's and Children's Hospital, an NHMRC and MRFF funded clinician scientist. He's the Deputy Director, Clinical Research of the Robinson Research Institute, the University of Adelaide, co-leads the Early Origins of Health theme in the RRI, and is a Principal Research Fellow at the South Australian Health and Medical Research Institute. The multidisciplinary neonatal research group that he leads spans basic science, population health, epidemiology and clinical research in the fields of system physiology, perinatology, paediatric medicine and public health. Thank you, Michael.
Hello everybody. As you can see, I've climbed out of my deathbed to come this evening. I hope I make it to the end. But it's just an illustration of how importantly the neonatal department take our participation in the Pridmore meeting, so thank you very much. I've never spoken here before. Most of you will have probably had to tolerate me at some point in my clinical sphere, but I'm going to focus this talk on my kind of Robinson hat, which is co-leading the Early Origins of Health theme within the Robinson Institute. So we're going to talk about the role of the placenta in developmental outcomes following preterm and term birth. And I thought I'd start with intrauterine programming. One of the best things about having an invited talk is the ability to have a tall tale. And I'm a little bit shocked, actually, having listened to Peter tell his story about having his picture taken with the Queen, to find out it was a picture with the Queen's portrait. But those of you that know a little bit about intrauterine programming will probably have heard the name of David Barker. He was at the University of Southampton. The last time he came to Australia was whilst I was doing my PhD in Newcastle, New South Wales. I'd worked the night before, I'd collected 2 placentas. I'd worked the whole day. I went to listen to his talk and I slept right the way through it. And that's my tall tale for the talk. With regards to programming, it really started off with quite a simplistic view. It was really about nutrients, really. And it's clear that the pattern of intrauterine development and size at birth are critical in determining life expectancy. And this kind of nicely brings us back to Brian Pridmore, I think, because this was based on midwifery held records of birth weight in the early 1900s, which showed that the smaller you were, the much higher rate of early cardiovascular disease, stroke you had, particularly in males. But the processes by which insults during early development lead to permanent changes in tissue morphology and function have been termed intrauterine programming, and clearly with the placenta being the main interface between the fetus and the mother, it's really central to this. And again, the simplistic thinking was there was 2 main ways. There was a supply of nutrients and oxygen required for fetal tissue accretion and energy metabolism, and then there was a production and metabolism of a range of hormones that affect fetal growth and development. But we've really begun to expand our understanding of the placenta's role in programming, not just hormone production, metabolism, or nutrient consumption and production, but also the role of transporters in the placenta, and I'm going to show you some of our work that we've done in Adelaide, the influence of size and morphology, which I think was really nicely shown by Peter, and again the role of blood flow. So the placenta responds to environmental stimuli, and it's been called a nutrient sensor. So in response to a modified nutrient and hormone availability, there's really 2 pathways. You take a fork in the road. You either, the placenta adapts and maximizes fetal growth and maintains pregnancy until it reaches viability, or you get a mismatch between the drive for fetal growth, which resulting in placental adaptation and programming. And as technology has raced away from us, we now know that this results in a whole lot of processes in the placenta which were beyond our original simple understanding. Gene imprinting, DNA methylation, histone modifications, altered mitochondrial oxidative function. And we need to remember that Adelaide has been at the forefront of most of these, particularly Claire Roberts, when she was at Adelaide, now she's at Flinders. Did a whole lot with Gus Decker out at the Lyell, with STOP and SCOPE, looking at DNA methylation and epigenome differences in the placenta. And it's clear that with the placenta having an important role in intrauterine programming, its phenotype might be a better marker of disease risk in later life than many of the other indices that we currently have to identify exposure to suboptimal intrauterine conditions. Really broad things like how heavy you are at birth. So it could be much, much better. An example, just to show you some of my work, this is one of the first publications that I had in Adelaide when I came back from the Queen Mother's in Glasgow, was to look at transporter proteins in the placenta, and something called PGP, which is a protein which shuttles cortisol back out to protect the fetus. We know that glucocorticoids are essential for the development and maturation of fetal organs, and that there's a clear rise in cortisol concentration during late pregnancy, which parallels increased maturation of fetal organs and the slowing of fetal growth. And what we showed was that in small for gestational age babies, there was a decrease in PGP in the placenta, which suggested that more cortisol got across the maternal fetal barrier to the fetus and was central to the pathogenesis of SGA due to a net increase in the exposure of bioactive glucocorticoids. The next thing I want to talk about is the evidence for sex-specific alterations in placental function and fetal well-being. And we, as a group, have always thought of the placenta as having a sex. It's a female placenta or a male placenta. And I did my PhD in placental biology. That's one of the problems for a naive clinician being supervised by a basic scientist. You get sucked into something that you really don't know what you're going to end up doing. And it's really interesting, the kind of interaction of sex and what it means for the baby and for baby's outcome. So clearly there's a major role for sex chromosomes. We know that there are differences in pre-implantation if you have female X expression. We know that there are a whole range of Y-linked gene expression disorders. You get histocompatibility antigens. Then you move further into pregnancy. By that I mean 8 weeks. And you begin to get the influence of the gonadal hormones, and particularly testosterone, which results in a gonadal female or a gonadal male. And what we were very interested in was how that impacted upon fetal development and the role of sex differences within the placenta on developmental trajectories across the second and third trimester of pregnancy. Because we know that there are clear sex-specific risks for adverse outcomes, particularly early pregnancy loss, preterm birth, fetal growth restriction, preeclampsia and stillbirth. We know that for, if I put my neonatal hat on, that if you're a preterm male, your rate of mortality is 10% higher. And for the male neonatologists, we've always rationalised that by, well, we're obviously the alpha males because we've made it this far. But it's clear that throughout pregnancy, sex differences arise from chromosomes, sex hormone biology, and that combined effects of both influence placental function, which may then influence healthy development. I think the take-home message from this slide is that basic scientists can't draw. This is a paper that we wrote with Fianna Morrison at UniSA. Vicky Clifton, who was here, is now at the Mater in Queensland. And it kind of encapsulates the differences we think between the male and female fetus and the relationship between the placenta and the fetus in male and female pregnancies. So for the female, you get increased placental reserve capacity. They have increased fetal placental adaptability, but the trade-off is a reduction in growth trajectory, but increased survival. because males are inherently not very clever, they maximise growth. You get reduction in fetal placental adaptability, placental reserve capacity is reduced. You might grow well, but unfortunately you've got significantly higher rates of intrauterine morbidity and mortality. So what underlies this kind of fundamental difference between the male and the female placenta? Well, as I said, I wasn't entirely sure what I was letting myself in for when I did my PhD, and this is data from the publication that I have with the most citations. It's just ticked over 200, which is about 0.5 of all publications will have a citation rate of that much. And what this does is looks at enzyme activity in the placenta, and particularly 11-beta HSD2 activity. And the reason for the citations are not necessarily the quality of the science, but the fact that nobody's stupid enough to do the experiment ever again. Because this was 6 weeks of my life in a sealed room, doing good old-fashioned liquid chromatography, using chloroform, which kills you, silica-coated glass plates, which you scrape the silica off, which kills you, and radioactive cortisol, which kills you. And what we showed was that in the placenta of preterm babies, these are all babies less than 28 weeks, in relation to time of delivery from maternal betamethasone exposure, and they presented in threatened preterm labour, there was a sexually dimorphic effect. So if you were male and you were delivered within 72 hours, the level of 11-beta HSD2 activity was significantly lower than females. So more cortisol got through to the male fetus, an effect that disappeared after 72 hours. And you think, well, why is that important? Well, actually, cortisol is one of the prime hormones that controls cardiovascular transition in the preterm baby. And I was very interested in how our prems born very early transitioned from being a fetus on the inside to a baby on the outside who can control their blood pressure. And what we found was the change in 11-beta HSD2 also related to a change in umbilical arterial cortisol and, more importantly, blood pressure and cardiovascular stability in the babies. So the males were much more likely to be cardiovascularly unstable. So it's clear that alterations that the placenta can make confer sex-specific advantages in neonatal outcome. And this I don't think will ever be repeated. So it's not just the glucocorticoid pathway though, it's probably also the androgen pathway as well. As I said, fetals prioritise pathways regulated by glucocorticoids to enhance placental reserve at the detriment of growth. So they slow down to survive. Males prioritise androgen-mediated signal pathways to enhance growth at the expense of placental reserve capacity and therefore increase their risk of intrauterine death. So in the presence of a maternal complication, the increased ability, adaptability of a female fetal placental unit, and I think we have to think of them as a combined pair, actually reduces their risk of intrauterine morbidity and mortality. And this has kind of generated quite a complicated diagram. This is work by Hayley Dickinson, who was at Monash, which tries to describe the potential critical windows for disease susceptibility in males and females and where changes in the placenta come into play. And as you can see, they're very different. You get changes in the structure of the placenta, as you very clearly showed in the first talk, of changes in the labyrinth, the vasculature, branching and proliferation of the placenta. Whereas females, you actually get a reduction in the labyrinth, the net result being reduced nutrient transfer, maintained viability, but severe fetal growth restriction. And it's very different between the two. And this is an area of work which has really been under-investigated because this is mostly done in a preclinical model. You can't do this in babies, so it's an animal model. Most animal models use one sex, and the majority of them are male because the females are used as breeding stock. So this has doubled the cost of all our preclinical work because we now have to have equal numbers of male and female offspring to fully investigate the difference in sex-specific differencing. The next bit I'd like to quickly go on to is, can the placenta actually provide insights into neurodevelopmental outcome? So for neonatal medicine, survival is no longer good enough. We have to ensure survival free of significant neurodevelopmental disability. And can insights in the placenta actually identify potential therapies? So this kind of focuses on the brain. We know that the developing brain is a nutritionally demanding tissue, and it's particularly sensitive to insufficiencies or overabundances of specific nutrients and growth factors, so kind of the classic intrauterine programming model. The CNS development is a process that starts very early, within 2 to 3 weeks of fertilization, but actual neuronal proliferation occurs much later in the first trimester, extends out into the second trimester, and then the really important bits of brain development, neuronal migration, synaptogenesis, are in late second and third trimesters. And this is a major problem for our preterm babies because they're disconnected from the placenta during this stage. So while the brain is extremely plastic during early pregnancy, it's acutely vulnerable to broad environmental fluctuations, and that might take the form of cortisol, as I've already shown you for 11-beta-HSD2, certain hormones, particularly serotonin and the thyroid hormones, but there's a whole lot of other stuff that may impact upon ultimate neurological outcome for the baby. We wrote our review in 2014-15, I think, which identified all the things that we thought were important that occurred in the placenta, which then altered outcome in the baby. It'll come as no surprise to anybody in the room that inflammation was right up there. We know that chorioamnionitis, particularly phenocitis and fetal inflammatory response syndrome, is a major player in poor outcome. But we got increasingly interested in some of the less studied pathways, particularly neurotrophins and neurosteroid synthesis, and then looked at how we could identify potential biomarkers, SNPs, MERS, DNA methylation, because they all impact upon brain development and result in motor, cognitive and behavioural deficits. And when you look at the literature as a whole, there's actually quite a lot out there already. As I said, histological chorio and phenocitis clearly is associated with adverse neurodevelopmental outcome, which is why it is just a no-brainer that all our placentas from preterm babies should go for pathology. As Yi and Peter have shown, vascular necrosis and fetal thrombotic vasculopathy also important, particularly for cerebral palsy, but also neurosensory deficits and motor disorders. But it's more the things further down the bottom which are areas of emerging research where there are particular differences in the placenta, particularly in SNPs, which have been shown to be associated with really quite significant adverse mental or psychomotor developmental index outcomes. The problem is you can't actually diagnose it whilst the placenta is still connected to the baby on the inside. And a lot of these things, unfortunately, the horse is bolted, and we're already dealing with the aftermath of changes that we can't reverse. There is, however, light at the end of the tunnel. One of the things that we were interested in, particularly something called neurosteroids, has been worked on extensively by Hannah Palliser and John Hurst at Newcastle, where I did my PhD. Neurosteroids are metabolized in the placenta from cholesterol, and the end result is something called allopregnanolone. And allopregnanolone is really, really important for the kind of advanced brain development that we see in the developing fetus. This is a model which has been proposed where understanding placental function can actually guide the development of new therapies to protect neurodevelopmental outcome. As I said, neurosteroidogenesis in the placenta and the developing fetal brain is critical for normal development. and the majority comes from the placenta, although there is capacity in the fetus to do it as well. And the loss of the end product of the pathway, allopregnenolone, results in a loss of inhibitory sleep-like state, oligodendrocyte maturation and disruption of the GABAergic pathway, and that results in a very unhappy baby. The cool thing is you can replace it, and this is now getting close to clinical trial. There's an agent called ganaxolone which does exactly the same thing, and there's now a large body of really lovely, very beautifully designed guinea pig work where preterm delivered growth-restricted guinea pigs treated with ganaxolone have exactly the same outcome of guinea pigs who are appropriately grown and born at the right time. And it's all really centred on myelination and how it results in normal brain development. So up until now, I've tried to be relatively sensible. I've tried to look at it from a holistic obstetric and neonatal midwifery point of view, but I thought I'd finish with the weird and wacky, and we'd go out to the final frontier. And this is whether the artificial placenta will actually become a reality. 2015, it seems like a long time ago because we're not allowed to travel anymore. Might have been 2016. Following one of the big American meetings pass, we went to CHOP and University of Pennsylvania in Philadelphia, where they are probably the group furthest ahead in the world with the concept of the artificial placenta and womb. And this is run by the leader of the group is Marcus Davey, who was at Monash and is now at CHOP. And I could not find the picture of this lamb subsequently standing on its mother's back in the paddock. So they've now got lambs going out, being delivered at 123, 124 weeks, which is about as low as we can look after a preterm lamb in a lamb NICU. Managed in the artificial placenta and taking them right out to adulthood. This has really been based on technological improvements which have resulted in a much closer fidelity to uteroplacental physiology. I'll show you in the next slide, but they started most of this work by building stuff in their garage, and the original pump that drove the artificial placenta was from an aquarium. So it was a little thing that oxygenated your fish, your tropical fish. And they're now beginning to show really quite impressive improvements in survival and hemodynamic stability, which has raised the possibility of more rapid translation into the clinic. And Marcus came to PSANS, which is the perinatal meeting in Australia, I think in 2016, and said CHOP would be doing this by now. They haven't done it by now because it's much more complicated than it made it out to be, but they're really pushing ahead. And it's not just the crazy Americans. In Perth, there's Eve, which is a very similar model. And from the original work, they're now taking lambs way down to like 105, 112 days, which is about as low as you can really go, and taking them right out to adulthood. And this is something which is coming, I think. This is like ECMO for the preterm baby. So I'd just like to finish by saying that the placenta deserves more than a shelf in the fridge. I collected them 24 hours a day, 7 days a week for 3 years and firmly fell out of love of them towards the end. And even though I said I'd never do it, we're actually coming back to doing it. And I wrote down when Amy put up the discussion points about cardiovascular disease in mothers. We now have a study running, Anna Kontos, who's at the Robinson, works for Declan Kennedy. We have a study looking at mothers with sleep disordered breathing who have chronic abnormal inflammation and vascular reprogramming. And we're beginning to show that it results in vascular reprogramming of the placenta and very abnormal smooth muscle vasculature of the umbilical artery and vein. I've carried boxes of placenta in tissue blocks around the world, back to the UK twice in each direction, and we've shown that those relate to changes in the baby. Unfortunately, I'm coming back to doing more placenta work. But it's clear that our understanding of placental adaptations in response to pathophysiologic pregnancy does remain rather basic. And while technological advances may increase our ability to quantify placental dysfunction, the wonders of ultrasound, placental MRI, it's likely that the impact on the developing fetus and therefore the newborn has already occurred. And we have to actually think about how we then change the journey that we've set the baby on. And I'd like to finish there with just my funding and my affiliations. Thank you.
Thank you.